genes of you and me. You're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello, and welcome to the 19th episode of Discovering New Advances, or DNA, a podcast that keeps you up to date about the scientific world in an easy-to-understand language. So whether you're a student in middle school or grandparent, you will easily be able to follow along. I'm Karen. On this episode, I will give an introduction lesson about the replication of DNA. For more information, please visit dnapodcast.com or email in at info@dnapodcast.com. This episode is going to cover how DNA replicates itself. DNA replication is in theory a simple process. Unwind the DNA and match free nucleotides to the open sides of the strand, creating two identical DNA strands from the one original. The most remarkable part of the process is how flawless it is. Rarely, and I mean rarely, are mistakes made by the enzyme matching the free nucleotides with the original nucleotide in the DNA strand. The question students always ask is a simple one: why? In this case, why does a cell have to replicate its DNA? A cell makes two copies of DNA because an upcoming phase of the cell cycle is mitosis, where the whole cell splits into two. In order for the cell to be able to split into two, it needs to double the DNA as one set of the DNA goes into each cell. Before I go any further, I would suggest going back and listening to the very first episode, which reviews the structure of DNA, as I'll be referring to the parts of DNA throughout this episode. If you're pro the structure, let's continue on. We have a brief overview of the process. Now let's break it down into steps. We start at the bonds of the base pairs. The base pairs are located in the middle of the DNA strand. The bonds that hold the base pairs together glue the two sides of the DNA together. Once these bonds are broken by an enzyme called helicase, the two sides of the DNA are separated. The location of the separation of the DNA is called the origin of replication. If this process started at one end of the molecule and worked its way all the way up to the other side of the molecule, it would take an extremely long time and cells would not be able to replicate quickly enough. Therefore, the breaking of the bonds occurs at many locations on the DNA strand simultaneously. The original nucleotides serve as the template for the new nucleotides being added. This is why the original strand of DNA is also called the template strand. This section starts with the replication fork, which is where the original strand of DNA is first unwound. Again, that's by helicase. To keep the DNA from binding back together with itself, single-strand binding proteins stabilize the original strands. That way it doesn't rebind to itself. Recall that the two sides of DNA run in opposite directions. One is the 3' to 5' direction, the other one vice versa. The strand that runs in the 3' to 5' direction is referred to as the leading strand, as it continuously adds nucleotides to the original strand. The other strand, the lagging strand, runs in the 5' to 3' direction. We can think of this as running backwards. DNA polymerase can only add nucleotides in the 5' to 3' direction, which is why the 5' to 3' direction strand is thought of as running backwards, because you're adding molecules that run the other way, because again, it's anti-parallel. The template strand runs the opposite direction of the new strand being formed. The 3' and 5' I keep referring to is regarding the sugar phosphate, the direction of which it's going. Before DNA polymerase starts working, RNA primase lays down a primer. Just like painting, the primer goes on first to ready the material. To add nucleotides to this original strand, the DNA polymerase, which again is the enzyme that adds the nucleotides, starts working right in the replication fork and moves away from it, adding nucleotides. Once the DNA polymerase runs out of the original strand to add on to, that fragment of DNA is complete. The DNA polymerase goes back to the replication forks and starts adding again. In this lagging strand, when a section of the DNA is done, it is called an Okazaki fragment, which is just fun to say, by the way, Okazaki. The Okazaki fragments are then linked together by the enzyme DNA ligase 1. So the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously in pieces, which are later formed together by DNA ligase 1. DNA replication in eukaryotes, like you and me, occurs during the S phase of the cell cycle. S stands for synthesis of DNA. Synthesis means to make, so this is a logical name. The synthesis occurs within the nucleus of the cell. A rule of thumb to remember is that DNA never leaves the nucleus. The products of this process of DNA replication are two DNA molecules. Each of these DNA molecules has one strand which has remained intact throughout this process. The other strand is the newly synthesized strand known as the complementary strand. This type of replication is semi-conservative, as half of the DNA is conserved from the original parent DNA, while the other half is newly synthesized material. Therefore, half of the DNA strand is old and half is new. These DNA strands automatically form themselves into helices upon synthesis, so you have that double helix structure you're used to seeing instead of just a straight line. Before we wrap up, let's review this process one more time. Helicase unzips the DNA by breaking the bonds between the base pairs, holding the two strands together. This happens at multiple locations on the DNA molecule. Single-strand binding proteins stabilize the original strands to prevent them from rebinding. RNA primase gets the two open strands ready for DNA polymerase to add to new nucleotides to it. In the leading strand, this happens continuously, while in the lagging strand, this happens discontinuously. The lagging strand synthesizes in pieces known as Okazaki fragments. The Okazaki fragments are bonded together by DNA ligase 1. This process produces two DNA molecules, both having one original strand of DNA and one new, making it semi-conservative replication. So I hope this was helpful in covering how DNA replication works. If you want to learn more about other genetic concepts, please visit dnapodcast.com. We have the transcripts on there as well as the regular audio files. If you have questions, you can contact us at info@dnapodcast.com. To keep updated with our social media, you can like us on Facebook, follow us on Twitter, both at /DNA Podcasts. And also be sure to subscribe to us on iTunes. That way you'll automatically get updates. You don't even have to search for them and download them yourself. And if you're already on iTunes, please rate and review. That's the best thing you can do to make us more popular. All the links I mentioned are on the website, dnapodcast.com. Thanks for listening, and join me next episode to learn and discover new advances in the world of genetics.